Welcome everyone. Good morning, good evening, good afternoon. Thank you for joining us today. This is going to be an exciting webinar. We have a lot of participants. Today we're going to talk about building a fair data platform for omics at a large pharma and how multimodal data is revolutionizing the drug discovery and development process. My name is Devika Garg. I'm the Director of Product Marketing at TileDB. I've been in the life sciences industry for over a decade now, both in bench science and product roles. And I'm thrilled to facilitate this session today. We have Maher Hamdi with us, who will make sure that our webinars go smoothly. Thank you, Maher. And then we have our wonderful speakers, Chad Krilo, Senior Genomic Solution Architect at TileDB, and Raghavi Shanmugam, Senior Bioinformatician at our partner Zifo. And they will take us through the story. We're so fortunate to have Chad and Raghavi today, who will bring a wealth of experience working in bioinformatics, particularly in multimodal data. And this will be interesting for everyone passionate about leveraging data to enhance healthcare outcomes, ultimately. I think anybody who's been working in this industry would say that, yes, in today's landscape, the integration of diverse data, health records, clinical data, multiomics data, clinical images, this is not just beneficial, but it is essential to drive effective decision-making throughout the drug discovery process in biopharma. And today we are going to talk about fair platforms and multimodal data and show you really how TileDB and Zifo have come together to implement a solution for this at a large pharma. To start off, I want to do a quick level set with a couple of quick polls. Merrick, if you could please throw up the polls up. And these are really to get an understanding, like how familiar are you with FAIR? That's an acronym thrown around a lot in the industry: findable, accessible, interoperable, reusable. These are principles for data management. We'd love to understand how everybody is familiar with this. And another thing, like, what is your biggest pain point with your current data platform today for managing omics data? Is it more around integration, interoperability, scalability? And we're going to go through all of these things in our presentation. Let's push the results. Okay, great. All right, so I think largely, like a vast majority are very familiar or somewhat familiar. And so that's great to know. We're going to touch upon this. And from a pain point of view, data integration, interoperability, and limited scalability, these are emerging as the key issues. And so I'm really glad. I think, Raghavi, you'll be excited to see this because we're going to address many of these things today. So let's get on with it. Without further ado, I'm going to step out. And Chad, please take it away. Chad, Raghavi.
Yeah, definitely. Hello, everybody. Really excited for the discussion today. If we could advance to the agenda, we'll get it going, please. Excellent. So in the area of precision medicine and complex biological research, multimodal data is no longer a luxury. It's really a necessity. So what exactly is multimodal data? Well, it's about integrating diverse data types like genomics, transcriptomics, proteomics, imaging, and clinical data to gain a more complete understanding of biological systems. And why is this so critical? Well, because relying on just one data type is like trying to diagnose a complex disease like cancer by looking at only one gene. Multimodal data gives us the complete picture, like having all the pieces of a puzzle. Well, so where is this really making a difference? Well, in personalized medicine, for starters, where we can tailor treatments based on a patient's unique genetic makeup, lifestyle, and environment. Drug discovery is another prime example where we can identify new drug targets and predict drug response. And in diagnostics as well, where we can improve accuracy by integrating imaging data with molecular profiles. Ultimately, multimodal data is key to unlocking new discoveries and driving innovation in healthcare and life sciences. There's also, as you all know, there are many challenges with multimodal data. While multimodal data holds immense promise, it also presents significant challenges due to the inherent complexity of dealing with diverse data types. Data variety is a really big challenge. We're dealing with a mix of everything from genomic sequences to medical images, clinical records, and more, each with its own unique format, structure, and scale. Data integration is another challenge. Bringing all this data together requires connecting these diverse sources and resolving inconsistencies. As always, data management is another major challenge. The sheer volume and variety of multimodal data can be overwhelming, requiring efficient storage, organization, and access. Data analysis as well. Extracting meaningful insights from this complex mix requires specialized analytical techniques and powerful computational tools. Finally, computational demands. Analyzing multimodal data often necessitates significant computing power and specialized infrastructure. But don't worry, exciting advancements in areas like cloud computing, machine learning, and data visualizations are emerging to help us overcome these hurdles and unlock the full potential of multimodal data, which is exactly what we'll be exploring today. So today we're diving into the incredible world of omics, where we're witnessing a true data revolution. As you all know, omics is all about studying life at the molecular level. Genomics, proteomics, metabolomics, you name it, and the amount of data we can gather in these fields is absolutely skyrocketed. Think back to the Human Genome Project, a monumental effort that took over a decade to complete. Now, with advancements like next-generation sequencing, we can sequence an entire human genome in a matter of hours for a fraction of the cost. This explosion of omics data is really transforming our understanding of biology. We can now uncover the intricate workings of cells with incredible detail, identify subtle molecular changes that drive disease, develop personalized therapies tailored to an individual's unique genetic makeup, and even engineer crops to be more resilient and nutritious. But this data overload also presents us with some really exciting challenges. On the next slide, we'll explore how we can effectively manage, analyze, and interpret this wealth of information to unlock the full potential. All right, let's see how this multimodal magic plays out in the real world. This diagram shows us how different omics technologies are applied throughout the drug discovery development process. As you can see, it's a journey with many stages, and each stage can benefit from multiple types of data. Early on in target identification and validation, genomics, epigenomics, and transcriptomics helps us pinpoint promising drug targets. We're looking at all these things like disease biology, tissue distribution, and gene expression patterns. Moving into preclinical development, we bring in proteomics and potentially even more granular transcriptomic data, think single cell or spatial transcriptomics, to characterize drug activity, understand potential toxicity, and select the right preclinical models. Finally, in clinical trials, we use a combination of omics to stratify patients, discover biomarkers, and monitor treatment efficacy and safety. This is where personalized medicine really shines. Notice how multiple omics layers are used at every stage. This highlights the intersection of these data types. The specific omics technologies used can be tailored to the research question and the drug modality. This integrated approach ultimately leads to more efficient drug development, better treatments, and improved patient outcomes. Now that we've seen the big picture, let's dive a little deeper. Okay. We recently conducted a survey of researchers like yourselves to understand the challenges and opportunities they face when working with multimodal data. One of the key takeaways was a clear tension between the potential benefits and the practical difficulties. This sentiment was echoed by many of the researchers we surveyed. They recognize the power of multiomics, but they also are struggling with the challenges of managing and analyzing this complex data. On the one hand, everyone recognizes that multimodal data fosters a significant competitive advantage. It allows for deeper insights, more personalized treatments, and faster drug discovery. But on the other hand, the complexity of this data can create a significant barrier. It separates scientists from the insights they seek and slows down the pace of discovery. Many researchers find multimodal data terribly complex to use with existing tools and infrastructure. That's where TileDB comes in. We've designed an ecosystem that simplifies the management and analysis of multimodal data, making it accessible for everyone. Okay. While frontier data like multimodal, multiomics data promises incredible scientific breakthroughs, as we just saw, it also presents significant challenges that researchers must overcome to unlock its full potential. So let's explore some of these hurdles. This slide highlights some of the common pain points we've heard directly from our customers in the pharmaceutical industry. These are hurdles they encounter every day when trying to extract valuable insights from their complex data. These challenges are not unique for any organization, really. They represent the shared struggles that researchers face when dealing with growing volume and complexity of multimodal data. But the good news is that solutions are emerging. The promise of multimodal data is undeniable, but it's crucial to acknowledge the hurdles that stand in the way of progress. Let's explore some of those roadblocks and discuss how we can overcome them. Okay, let's be honest. Working with multi-omics data can be a real headache sometimes. From data silos to access requests, there are plenty of obstacles to navigate. Here's how we address those struggles. TileDB breaks down data silos by unifying diverse omics data types in a single scalable platform, so no more fragmented views. TileDB streamlines data access with efficient querying and built-in security features. Researchers get the data they need when they need it while maintaining data governance. TileDB also prioritizes data security and compliance. Our ecosystem helps manage consent and access control, ensuring your valuable data remains protected. TileDB empowers your bioinformatics teams with a high-performance ecosystem that simplifies data management and analysis, frees up their time for discovery, not data wrangling.
This is related to the question that we had earlier, but a little more digs a little bit deeper into scalability, privacy, combining data. If we can please get a sense of some of these deeper dive struggles. And really like what Chad was highlighting earlier, we've done a ton of market research and across biopharma, and these are the themes that have emerged for us. So we'd love to understand. And let's run this poll for perhaps 30 more seconds there.
I mean, during my time outside of TileDB, when I was a research scientist at the NIH, I dealt with these topics on a daily basis, right? Not every single one every day, but they always came up. You know, these were really the major ones that I encountered for years.
I'm sure like Raghuvi, you'd see that like on your side of it, things.
Yeah, we hear from these exact challenges from various customers.
Okay, so we're seeing, I think that's interesting, like combining data, again, interoperability from disparate sources, because a lot of data has been generated in-house, some public data, data in CROs. All right, great, thank you. Let's keep moving. And Raghavi, I believe you're going to take us through integration.
Thank you, Devika, and thank you, Chad, for going over all the multimodal challenges. And thank you, audience, for the poll results, because as the major answer points out, we are going to talk about how we are going to combine data from disparate sources. So this slide, if you as an organization working on a multimodal data challenge, will be very, very relatable to you, because once you jump into this data strategy for multimodal integration, you will be like bombarded with lots of questions like where the data is coming from, what are the data sources, how is it being governed, what compliance rules apply at each data layer or each data source, how are you going to integrate the data, how are you going to store the data, because there are plenty of options available. Along with TileDB, there are a lot of vendors who are also like looking at multimodal data challenges, and each one having their pros and cons. And then even if you store, govern, clean the data, everything is good. How are you going to present it to the consumers? That emerges as the major challenge for most organizations. And if not just presenting the data, how are you going to best utilize the data in this AI era, right? Everyone is jumping into this, AI, ML prediction models and methods. And how are you going to best utilize this data? And how are you going to develop a strategy for it? And which data are you going to use? So there are like a lot of questions that that are sometimes not answered. So this is where I think to clean your data and to govern your data, the FAIR principles really help you. So the FAIR principles, I would say, is more like a checklist to tell you how are you going to store the data, whether you are taking care of all the important aspects of data governance, and all the important, whether you have got everything covered for the data strategy, right? Devika, next slide, please. Yeah. So this is the high level FAIR principle. What it says is findable, whether your data is findable, whether it is accessible to the right set of users, whether you can like ship the data from one system to the other system, whether you can reuse the data efficiently. So these are the major aspects of FAIR, and there is also like one more added in the recent years for reproducibility, whether you can reproduce the insights again and again. So again, this is not a hard and fast rule. So it depends on your data strategy and where you as an organization stand in terms of digital maturity, and what is that you're using the data for dictates, how many FAIR principles you're going to apply to your data pipeline. Next slide.
Yeah,
Yeah.
again, I want to understand, right? That wherever you are in your organization, whichever organization you're with today, where are you in that journey of achieving FAIR? It is a very lofty goal, ideally to be able to use data, all that data that is being generated, to be easily discoverable. So important as a foundational competitive advantage. Raghavi, I'm curious, like how often do you see, in your experience, companies coming in asking specifically for that, hey, we're here, please can verification is the first thing that we want to do before anything else?
It's very often, and sometimes when you talk to them, you can see where they are in terms of verifying their data. And most often, not to our surprise, it's very, like, I would say we are in the very initial stages of this data revolution, because, we are generating a lot more data, and we don't have any like a golden rule or standard to say this is the way of storing and governing your data, especially when it comes to omics, right? Multiomics problems. So yeah, the verification is, I would say, the number one question we ask our customers when they are starting to set up their data pipeline.
And it's not just at the data generation step, but also throughout it. Like, can you track the analysis on that data, which goes on to that reproducibility deeper? Like, once you've created the data in the lab, it is going through a journey of analysis. And so can you track and audit all of that? All right. How are we doing? Should we close and push results? All right. Yeah, I think
This
comments here.
Yeah, this is such a divided poll. There is not a single answer. I can see, yeah, this is something that we see in the real world use case when we talk to our customers. Each organization is different, and also even within the same organization, depending on which team you are working with, says where you are with respect to FAIR.
Yeah, I'm actually not surprised to see these results. Honestly, I was expecting to sort of see them across the board. Throughout my time before TileDB and at TileDB here, working with a lot of pharma companies, some people are starting to explore FAIR principles. Others are fully FAIR compliant. A lot of the folks are sort of in between the two, right? Actively working on implementing FAIR, right? Especially when you're talking about the reproducibility aspect, right? Anytime there's like FDA approvals required or things of that nature, you need to be able to reproduce those end results, right? And if you don't have code, data fully available and findable, it's going to be super hard to have that reproducibility on the back end, which is of the utmost importance, really, I think.
All right. Thank you, everybody, for that.
Yeah, let's jump into the use case. I think all the polls that we had during this webinar and the results indicate that everyone is curious to know, or at least looking into the multimodal data integration. So that's why we are excited to present today the interesting use case that we worked collaboratively with TileDB in the recent past. So we were presented with this use case earlier this year, trying to combine the clinical data with the genomic data and then present it to the users by overlapping it on the protein 3D structure. So this was the high level use case, but if you have to break it down, this had like so many layers. Like, how are you going to tackle this from the data perspective? How are you going to tackle it from the data governance perspective? And how are you going to look at it from the technical perspective? And also from the design perspective, because you can design a complicated application. If the users are not going to use it, then it's of no use. And then the most important aspect is the scalability perspective, because each clinical trial is different, and whenever the genomic data is in picture, the scalability is one of the major issues we have when you're fetching the data and displaying it. And on top of it, we have the technical challenge of rendering the protein 3D structure and also overlapping it with the genomic data. So it was altogether, I would say, the most interesting and the fun project to work with, because as you can see, it kind of tells you um, how we can demonstrate the value of data if you can integrate it from various sources. Because even though the data is siloed in your organization, in reality, the data, everything works together in the real world, right? So that is where demonstrating the real-time use case of data integration is really, really interesting for the scientist, and it makes the scientist to even look at the data in a whole different perspective. So as you can see, our data sources were, when I said the clinical data, the genomic data, and the protein 3D structure, all three buckets are very evident. But in order to combine and integrate them, you obviously need some supporting data sources. So that's where the ontologies come into picture, the public annotations come into picture, the PDB, the protein data bank as a data source come into picture. Then we have UniProt, and we also had Ensemble. So we need all these additional data sources to support this data integration in the transformation layer. So as you can see, the first box just lists down all the data sources. And in the second box, it shows how the data flows and how we can combine the clinical with the variant data and the protein structure. And we also have some IDs listed out there. Then comes the integration layer, where all the IDs, all the metadata needs to talk to each other, and and then we know which data and how the users are selecting the data to visualize in the end. So, and the another interesting point from the design perspective is the app was made to cater wide range of users. So it should cater the clinical biostatisticians or the clinical groups, the bioinformaticians, or even the target discovery scientist. So if you have like wide range of end users, then your design should be simple, at the same time complex enough so that they can dig into all the details of the data in the backend. Next slide, please. So this is just taking a step back, how the data is going to come into this application. So as you know, whenever you have a clinical trial, it starts from the clinical side. The data is procured in the clinical side. It goes through its own journey before it lands into, for example, the pharma organization, right? So in that first row here, it shows a single clinical site where the sample is extracted, it goes through the sequencing process, and then, you're probably like getting the data from your CROs or vendors or sometimes the public sources. And then at your organization, if you are, sitting in a pharma or biotech, then you're bombarded with, again, lots of data from lots of clinical sites around the world. And then, sometimes they are not in the same format or not processed in the same way. And sometimes, if it is coming from Europe or, a different country, there are different complaints rules apply with respect to how you're sharing the data. So there are a lot of complexities before even sourcing the data, right? Once the data came to your organization, you have to make sure it's all cleaned up, harmonized, and we are starting from a single point, like, right? It's not like, again, we don't want to go back and start from like primary analysis of data. So for designing this application, we had to like make few assumptions, and we have to make some rules to make sure that, we can rely on the data. So this is where the FAIR at source is one of the main principle we took in our hands to make sure that the data is stored when we start with. For example, if you take clinical data, we wanted to make sure that clinical data is all like in the same format. So one of the rules we had was all the clinical data must be in STTM format. Then when it comes to VCF, Chad will probably dig deeper into this. So when it came to VCF, again, that could be like multiple VCF versions, that could be multiple references, that could be, again, different processing methodologies. We wanted to make sure the VCF is all stored in a certain way, so we can rely on that VCF data. And another thing is, when you're talking about 10 patients, it's you can search from the file directly. If you're searching for 1,000 patients, maybe you can use something, like Tabix or some kind of indexing system to, scale the data. But if you don't know how many patients you're going to expect in a clinical study, then we need to make sure you're storing it in the right way so we can trust the performance of that system, right? That's where the TileDB really helped us, because it scales irrespective of the number of patients. So we can say, be it 10 patients or 10,000 patients, we can keep the performance time well under 10 to 15 seconds or something like that. Next slide. So now we talked about how the data should be at the source, then comes how we are going to integrate it, right? So another challenge whenever you're dealing with clinical data is you cannot duplicate or store the data everywhere, because it comes with its own access and security issues. That's a whole different detailed topic. But I'm not going to go into the details. So for this application, just take the underlying rule that, we should not duplicate and store the data from clinical studies anywhere else. So that means we should not have a persistent layer. Rather, we should be working with the microservices layer. And the microservices layer should be like querying data in real time from all these sources and then present it to our UI layer. So if you look at the slide, it says how the data is integrated in real time. So what happens is the user inputs a particular clinical study or a phenotype or a patient ID. So that is the trigger to start the process. So once the user enters a patient ID or a clinical study ID or a particular phenotype, then we go back and from all these systems that we talked about, like for example, from UniProt, we need to get the sequences, we need to get the transcripts. And another interesting aspect was to display both canonical and the isoform, all the structural variants of the protein. So we were getting all this information from UniProt and PDB. And then in TileDB, as I mentioned, we can query for all the variants associated with that particular patient. So we can get the list of variants. And sometimes we are not just looking at a single patient, it's list of patients from, based on the phenotype. So that means if you have, again, 10 or 100 patients, we were like going into TileDB to get that list of variants. So once we have the variants and the protein 3D structure, it's probably it was an MMSF file, and then the UniProt. Then we had the UI design to display all this information in, I would say, 3 different categories. So the first tab, if you remember, I said it was supposed to cater different set of users, right? So we had a dedicated tab to interact with the clinical metadata, because if you work with STTM data, you'd know it's not a very straightforward file structure to just say, okay, I want this particular measure to be within this certain range or something. So we had a dedicated UI layer to interact with the clinical metadata, so you can select the list of patients. Then we had second tab, what we call as 2D viewer, where you have the 2D protein sequence, and all the amino acid changes are plotted in there. So if you zoom into that 2D viewer, you can see how many patients have that particular amino acid change, and everything is, it's shown as I would say, like a 2D plot. Then we have the most important and the interesting layer, which we call as 3D viewer, where you had the protein 3D structure. We used Moldstar embedded into our application, and then the protein 3D structure files were sourced from PDB, and we included both canonical and isoforms. Then we also had computational and the experimental structures for the PDB structure. Then based on the TileDB variant positions and the UniProt and the ensemble transcripts, we were able to map the amino acid positions on the 3D structure. So when you are in the 3D structure, you can like interact it in real time and then see where your variants are. Because sometimes, in 2D, the variants could be like far apart, but when the protein is folded, it can, well within that target binding site. So that was, I believe, the interesting takeaway for our scientists. So this is how the data challenge was tackled within this use case, and then how we integrated the data. Yeah. I request Chad to dive deeper into the scalability aspect of and how TileDB helped us.
Yeah, no, thanks a lot, Raghavi. And that's really some fantastic work your team has done. Up until this point, we've talked about the complexities of multimodal data. And one of the biggest hurdles is, as Raghavi had alluded to, is just the sheer volume, right, and scale, especially with genomics. But it's not just about size, right? It's about sort of the speed that you need to see out of applications like this, right? We need solutions that work in real time, whether you're analyzing 100 samples or 100,000. The response should be comparable. So how do we tackle this challenge, right? Well, TileDB, of course, instead of relying on traditional VCF files, which can be cumbersome and inefficient for large cohorts, we use TileDB to model VCF data as analysis-ready arrays. Think of it like this, right? We're organizing the genomic data in a super smart, multidimensional way that makes it much faster and easier to access and analyze. TileDB's really secret weapon here is its super efficient engine. It's cloud optimized and achieves really unparalleled performance for genomic data, thanks to some key features, really. TileDB can efficiently store any number of samples in a compressed and lossless manner, ensuring the data stays compact without sacrificing any accuracy. You can rapidly add new samples to the existing data sets, eliminating the so-called N+1 problem and scaling out linearly, as Raghavi touched on earlier. This is really crucial for dynamic data sets that are constantly growing. But it doesn't really stop there. TileDB also harnesses the power of distributed compute in the cloud, allowing us to analyze massive data sets like those from biobanks and genomic atlases with incredible speed and efficiency. The result, well, really no more performance bottlenecks. TileDB enables concurrent analysis at biobank scale without sacrificing that speed and efficiency. As you heard earlier, you don't want to go to lunch in the middle of your analysis. Really, with TileDB, you can get your results quickly and keep your research moving forward. Next slide, please. Excuse me. Now, let's talk about how the platform, really the ecosystem that brings us all together, TileDB. It's not just for genomics, right? It's designed to handle the full spectrum of multimodal data in your research and development. As you can see, TileDB is built on 3 core pillars: data, compute, and governance. First and foremost, TileDB can handle all of your data, from genomics and transcriptomics to biomedical imaging and beyond. It's not limited to specific data types. It can accommodate tables, files, metadata, you name it. Next, TileDB provides the compute power you need to analyze this data at scale. We're really talking about distributed data pipelines that can handle massive data sets, along with tools like dashboards and notebooks to streamline workflows. And of course, it's all cloud optimized for maximum efficiency. Finally, TileDB addresses a crucial aspect of governance. It provides robust features for data sharing, authentication, access control, and cost management, ensuring data is secure and your projects stay on track. So in a nutshell, TileDB gives you a unified ecosystem to manage, analyze, and share your multimodal data, empowering you to accelerate your research and drive innovation. So really, now we've seen the challenges of multimodal data. Now let's look and dig into how TileDB's unique architecture addresses those challenges head on. Next slide, please. Okay, so this diagram provides you a high-level overview of the TileDB ecosystem. As you can see, it's designed to be flexible and scalable, accommodating various data sources and deployments. TileDB can seamlessly integrate with a variety of external data sources, including existing data lakes, cloud storage, and much more. This ensures you can bring all your data together in one place. For example, TileDB offers efficient tools to load and process VCF files, transforming them into analysis-ready arrays. At the heart of this system is the TileDB engine itself, providing the core functionalities for data storage, management, and analysis. TileDB leverages serverless computational infrastructures to provide on-demand scalability and cost efficiency for your data processing needs. TileDB also includes features for data cataloging, making it easy to organize, discover, and manage your data assets, making them fair. Robust security and governance features are built in, ensuring your data is protected and is access controlled. And TileDB offers many APIs and a user-friendly console for seamless integration and management into your data and workflows. We also facilitate collaboration, data sharing, enabling researchers to work very, very effectively. Architecture is really designed to handle data at extreme scale, from terabytes to petabytes without compromising performance. Ultimately, TileDB empowers various data consumers, including researchers, analysts, and applications to access and analyze all their data efficiently. At the end of the day, you have control of your data storage with the ability to utilize your preferred storage solutions. So this modular and scalable architecture allows TileDB to adapt to your specific needs and provides a robust foundation for your multimodal research. Next slide, please. Okay.
Back to the use case. Thank you, Chad, for diving deep into TileDB. A quick recap, we were talking about the bringing in clinical and VCF and protein data together. So this is the reference architecture we had. So we had the clinical data lake, which was designed to cater all the again access and security issues with respect to the clinical data. Then we had external data coming from TileDB, PDB, and we had another vendor for public annotations. So we had the microservices layer. Then we did have cache to store the user sessions and anything other than clinical data. Then we had load balancer, the certificate manager, all these things. And then we had what helped us was the ontology management system. So this is in terms of FAIR really important because to make sure when you're calling out a gene name versus gene ID, it's all harmonized and we are talking about a single gene, because to avoid any confusions with the gene list. And also with the clinical data, there could be like, some terms that are not matching with the other terms. So to harmonize the clinical metadata column names and all the stuff, we had the ontology mapping, ontology management system, so that helped us. And then we had the user access. So all this was helping us to design that application. So this is a very high level architecture, and if you have any detailed questions, we can talk definitely more about it after the webinar. Yeah. Next slide, Devika, please. Yeah. So I talked about FAIR principles and why it's important. Our customer was, when we were approached for this particular project, Devika asked me a question: how often do your customers ask for FAIR, right? Our customer had a very great understanding of FAIR, and when we were approached to design this application, they specifically asked us to make sure it's following all the FAIR principles. So once the development was complete, we did turn to the FAIR principles, and we did evaluate to make sure we are keeping everything covered for FAIR. So this is our FAIR evaluation metric. So against all the FAIR principles, we were like trying to make sure that the application follows these things, and these are the criteria that we met in this application. I'm not going to go deep into this. Again, if you have any questions, we can definitely address towards the end of the webinar. Next slide. Yeah. So I have few screenshots. I won't be able to demo the application, but you can see the screenshot of the clinical tab. So we had 3 different tabs. So the first one was the clinical tab. So our rule for the clinical tab was to have the data in STTM format. So the STTM format is again like little tricky. You have like observation, you have like lot of variables. So if you have to like filter a list of patients, then it has to go through like different layers. So we had a very nice feature to filter the patients based on various criteria. And then you can see like how once you filter the patients, you also get a summary of how many patients are there, how many total patients, and how much you have. Next slide, please. So yeah, this is another screenshot of our clinical filter page. So this just shows the clinical trial inclusion exclusion criteria, and you can see all the variables, and how you can like add more filter. And sometimes you will have like lot of filters. I know we can't store the data, but we can store the queries. So we have stored queries for the users, and then you can download the filtered data on the table that you have. So we have the option to download data for CSV and Excel. And another interesting part is not all the users will have permission to download the data. So even that was managed at our user access layer. Next slide. Yeah. So this is our 2D viewer that I mentioned, where we'll have the 2D protein sequence and then plotted with all the VCF data. So you can see the amino acid changes. On one side, you have all the list of amino acids, and on top you have the protein sequence, and you can zoom in, zoom out, you can see, and each circle here represents variant change. And if you click on that circle, you'll be able to see how many patients are having that particular amino acid change. We have consolidated all the frame shift into a single row here. So if there is a particular variable region that you're interested in, you can see in that first row. But when you click on that frame shift legend, you can dive deeper into how many frame shifts have occurred in this position and how many patients do have this. So this really gives you basically a deeper dive into data. This helps you interact with the variant data a little bit more. Yeah, this is the pinnacle of the application where you have the protein 3D structure. On the right-hand side, you have all the information about the structure and how many structures are there, whether it's a canonical structure, isoform structure, how is it being, how is it being rendered, whether it's a computational structure or the experimental structure. You have all that information on the right hand side, and then you have the option at the bottom to select a particular chain, or you can zoom to a particular variant, because if you have hundreds of variants, it's very difficult to look at the variant of interest. So you can filter the variants. You can either filter out of thousands of variants, you can filter probably to 5 variants. And even within the 5 variants, if you're interested in zooming into a particular variant, you have the option to zoom into that variant towards the bottom. So this was really fun to even look at. Yeah, so this shows how you can basically bring the data together and make it more interactive to the users, so the users get a whole new perspective of your data. And yeah, this opens a lot of possibilities. So along with this, another option I didn't mention so far is you can even upload your own PDB structure. So for example, if you're doing your own prediction models, if you're like trying to work with the complexes and trying to come up with your own structure, you have the option to even upload your own structure along with the publicly available PDB structures.
This is really so wonderful, and so many scientists are using it. We have a really nice surprise here. We have with us Marcin von Krothus. He's the Director of Data Integration and Analytics at Takeda, and he's joining. Hi, Marcin. I hope.
How are you? Can you hear me?
Yes, we can hear you. So this project is a Takeda project, and Marcin, we'd love to learn like a little bit more from your end. Why was it so important for Takeda to do this, and how some choices, and how is it impacting?
Yeah, thank you so much for giving me a chance to share how this looks like from our end. And apologies, I don't know how to turn on the camera. Probably it's not possible to do it for the webinar. But this is what I wanted to tell you, that this entire journey was amazing from 3 different perspectives. First of all, is the product that we got at the end. It's a very unique product because it simultaneously joins 3 different types of the data: the patient level data, the clinical data, the genomics data, and the public available data. It's the kind of the first time you have the possibility to on the fly select the patient, see what the patient have the phenotype, then go to the TileDB, extract on the fly the patient genomics variants, translate those genomics variants to the amino acid changes, and map the patient's phenotype on the 3D structure of the proteins, with the first time opportunity to see whether the amino acid changes that colocalize in the 3-dimensional space of the proteins, do they correspond to the same patient's phenotype, like the age of the diagnosis, the severity of the phenotype, which not only could like help us to better understand the mechanisms of actions behind the genomics variants in the 3D space of the proteins, but also it may point to the druggable regions of the proteins that could be used for the future drug discovery. That's one aspect. The second aspect of this entire journey is that this partnership with Zifo and the TileDB and Takeda was really great, because in a very close period of time, Zifo was able to bring a big team that could like implement this very fast. Then we could use off-the-shelf product being available that was provided by TileDB, so we didn't have to do everything from scratch. It's give us a huge speed at the beginning, and then from our perspective, we could nicely join those 2 things together for our own needs. And the last but not least, is what's like really amazing was to see how Zifo teams grows within this journey and how they become better and better and faster delivery a new request, and also seeing how TileDB changed their product in the same time dramatically. Like when we joined the partnership with TileDB, when you're uploading the VCF files, it has to be like one sample per VCF file, and it was kind of slow. The upload was kind of slow at the beginning. The speed of the retrieving the variant was unbeatable from the beginning, comparing to all other uh, similar solutions. But during the entire journey, now you can bring a single VCF files for millions of the variants and thousands of the samples, and within a few hours, it's uploaded right away because they utilize the parallel upload, and it speed up the entire process. So from all those three angles, it was like really great to do this project together.
That is so wonderful to hear. And like, I'm curious, what is the uptake looking like within the organization? And I think many of the folks who are listening in, I think it would be interesting.
So our next step is to expand this to beyond the just genomics data and go to the single RNA-seq data, to special transcriptomics data, and to other data modalities. And with Zifo support, go from this, on one hand, amazing solutions, but expand this to and scale it up to our other modalities and ask other scientific relevant questions from the perspective of the target identification and validation.
Got it. Got it. Did we want to talk any more, Raghavi, around these pieces? I think something like Marcin, you already alluded to the multiomics integration and adding more data, upstream, downstream applications. Anything else you want to touch over here, Raghavi? Or
I can add that we are working right now on a new big proposal, how to expand our collaboration to the next one level. And if you're interested on bio IT, I will be giving a talk when I will be in the same time advertising TileDB, but from our perspective, showing how amazing this tool is for our own purposes.
wonderful. This is so great, and I think the joys of a live webinar. I love it. Thank you so much, Marcin, for stepping in and sharing your perspectives.
Thank you so much to TileDB team, and thank you so much to Zifo team. You know, without you, we couldn't have this great product so quickly developed, and we are looking forward for our continuous partnership. Thank you.
Thank you again. Thanks for joining.
Mhm.
And Raghavi, do you want to bring this home with a summary?
Sure. Yeah. So yeah, Marcin gave a nice summary about how this application is being used in his organization. But in terms of development and the challenges that I initially addressed, so this application is used to demonstrate three different elephants in the room, which is the data integration challenge and the scalability challenge, and bringing in the whole ecosystem together. It's not just about data. It's not just about the technical challenges. It's not just about the end user. It's about bringing everything into the same plate and making sure it is presented in the right way to the end users is very important. So yeah, having the right ecosystem and then having the right stakeholders and having a champion like Marcin who translates what the users want was very important for the success of this.
Absolutely. Thank you so much, all three of you, Raghavi, Chad, Marcin. There are a couple of questions. We're almost out of time, but one of them is like, are you seeing more pharma companies wanting to build this type of their own integrations for these type of use cases? And why do you think this is not an off-the-shelf solution?
Yeah, I can take a little bit of this and then sort of pass to Raghavi. And I think the answer is yes, right? with the people that we're working with, we are seeing companies trying to build their own integrations on top of these use cases, right? But then they ultimately hit like a bottleneck. They hit a wall, whether it's on performance, on querying, or the data is just too large, right? And it's not practical to have it in an S3 bucket, then copy it to an EC2, and then run analysis on that, right? So that's really where I think TileDB really shines in this question here is, our system allows these sort of bottlenecks to just go away, right? Yeah. What are your thoughts on Raghavi?
Yeah, so performance is one thing. Again, the decision to go for a off-the-shelf solution versus a custom solution really depends on their strategy, their short-term versus long-term strategy, and what are they envisioning for their data, right? I think one concern to not go for off-the-shelf that we see is they don't want their data to be held captive within the system. But I think in this cloud era, it's changing slowly. So people are more open to try out off the shelf solutions, and especially since it's SaaS solution, they can easily like transition from one to other. So there are like, again, pros and cons towards why one would go for off the shelf versus custom solution, because the custom solution definitely takes lots of effort, and yeah, the ROI should be justified if you are going to build a custom solution.
Absolutely.
If I can add
Wonderful.
something to this one from like
Yes, of
our
course.
perspective, it's like off-the-shelf solution is always preferred choice unless it really does not check all of the boxes, essential boxes. It's faster and it's cheaper. That's basically period. Because TileDB can sell the same product to multiple pharmaceutical companies, so obviously it's gonna cheaper, and we don't need to like maintain and develop the code, and so it's also significantly faster because we are already grabbing a tool that was tested and debugged by others.
100%. Yes, absolutely. I want to thank everybody again. Marcin, thank you so much. This webinar just became that much more exciting with you with you here. Chad, Raghavi, thank you so much. Maher, thank you. And I want to end with announcing our next webinar, which is going to happen on December 12th. Marcin teased this like single cell a little bit. And this is going to be about training ML models on Atlas scale single cell data sets. This is happening December 12th. Please do scan. We'd love to see you there. And thank you again for joining. We'll see you next time.
Thank you. I'm looking forward to.
Absolutely. Thanks,
Thanks,
Marcin.
Raghavi. Thank you, everybody. It was a pleasure.
Hi, everyone.